Welcome everyone to the 2024 Seasonal Update webinar for canola. This is brought to you by the Australian Fungicide Resistance Extension Network, also known as AFREN. I'm Anna Krieger, and as AFREN coordinator, it's my pleasure to bring you Dr. Steve Marcroft from Marcroft Grains Pathology. Now, Dr. Marcroft will be presenting vital information today on the economics of blackleg fungicide application as it relates to upper canopy infection in canola crops this season. For those who are new to joining us, AFREN is a GRDC-supported initiative that provides up-to-date resources and information directly to Australian growers, agronomists, and advisors. You can find these resources, including a fungicide resistance management guide, regionally specific workshops, fact sheets, webinars such as this one, podcasts, and more at our website, afren.com.au. Now, before we begin, a bit of housekeeping. If you have any questions during the webinar, please drop them in the Q&A box that is on your screen. We will aim to address any queries at the end of Steve's presentation. I'm pleased to hand you over now to Dr. Steve Marcroft, who is one of our leading experts in canola and blackleg disease here in Australia. With decades of experience in this field, Steve is well equipped to provide you with valuable insights. So I'm going to now hand you over to Steve.
Thanks, Tree. So as Tree just said, we're going to really go through what are the disease implications right now, standing in a crop in August, and then try and make some decisions about whether we need to apply fungicides or not. So firstly, I just want to acknowledge this is a national project. It is funded by GRDC. It's been running for many years, and the current investment is over the next four or 5 years. So just to sort of set the scene, I sort of thought I'd start with saying, where are we at right now with blackleg? And then go through a few of our genetics and then management, I guess, advice that we have in general, and then finish off with where we're at. So we've got about 30 slides to get through, so it shouldn't take too long. So obviously, everyone knows blackleg, devastating disease of canola. It's wiped out the industry in the past. However, right now, we're actually in a really good place with blackleg. And the reason for that is we do have a really good understanding of the pathogen. So we've been working on this disease for a very long time now, and we do have a great understanding of it. But I think the big advantages that we've got is that we've really got good plant genetics. So I guess over the last 20 years, with all the private investment, private seed companies coming in, we've seen a lot of diversity in our genetics for resistance, and that's really come through to the cultivars we've got now. So we're seeing basically cultivars which can tolerate a lot more disease than what we would have had in the past. And as you're all aware, which is why we're here today, is that we've got lots more fungicide options. So it wasn't that long ago we had very few options, but now we've got a range of options, and they're vastly improved as well. I think one of the big changes we've seen in the last few years is the importance of sowing and flowering time, and I'll talk a little bit about that and about how it affects the disease and how it changes how much disease we might get and how we put our fungicides on. So when we put all these things together, we've seen the canola industry, quadruple in size, which we, 20 years ago would have thought would have destroyed our industry from a blackleg perspective. But at the same time as quadrupling the size of our industry, we've actually now grown crops under a lot less disease severity than what we did 20 years ago. So it's a real testament to the technology that we've now got on hand. So that's a great situation. On the flip side, I guess the cons is that as I said, we know a lot about our pathogen now, and we know that it continues to evolve against our cultivar resistance. And that's just something that we're going to have to live with. It's something why we're always going to have to invest and always change cultivars and change practice, etc., because the disease is continually evolving against cultivars. As I said, we've still got quite a bit to learn about how to best manage upper canopy infection. We're getting there. We've got some real great breakthroughs, and obviously today one of the main reasons we want to have this talk is that we've just released the blackleg ratings for upper canopy, which we haven't had in the past. So that's a real step towards managing upper canopy blackleg. One of the things that we've become very aware of is that fungicides are fantastic. They always control disease, but the disease doesn't always cause yield loss. And that really comes into how do you then decide when the disease is going to be severe enough to cause that yield loss, and therefore you get an economic return from your fungicide. And as always, our change farming systems have had a massive effect on the disease, in some ways very positive and in some ways negative. So we'll touch on that as well. So, like I said, the really exciting news and the brand new stuff is that we've just released the blackleg management guide, the new one with the upper canopy blackleg ratings on it for the first time. And that's available on the GRDC website. So that was only released last month. So it's been a real missing part of the puzzle, I guess. We've had obviously blackleg ratings forever, but we haven't had any blackleg ratings for upper canopy. So people have been really stabbing in the dark there. So really, really happy that we've now managed to get that out. And that's obviously been the work that we've done with this GRDC funding, but it's also all the seed companies have come on board and supported that and have been happy for their cultivars to get labelled with the upper canopy blackleg rating as well. So it's been a real industry effort to achieve that. So starting now, sort of blackleg 101, one of the things we've realised over the last few years is that blackleg comes in different forms, and really in Australia now we talk about crown canker, upper canopy and pod infection, and really the two that we try to manage are the crown canker and the upper canopy infection. And really I really like to almost treat these as two separate diseases now. So the first thing I want you to be aware of is that with these diseases, they all originate from last year's canola stubble. So you can see there the stubble's loaded with fruiting bodies. Every time it rains, the ascospores get released. When the crops are small, they land on our cotyledons and our first leaves, and then they grow in the vascular tissue down to the crown and cause a crown canker, which you see at the end of the year. But it's all the seedling infection, which results in crown canker. Infection later in the crop doesn't result in crown canker. And then later in the year, so this time of the year, when our crops are coming into flower, we see the flowers being infected and stems and branches being affected, and the disease can grow along the peduncle into the stem. And again, it causes the same necrosis as we see at the crown. So the same process is exactly occurring, but they're just occurring at different growth stages. So the first thing when we're looking at blackleg is that in most situations, it's unlikely to get both severe crown canker and both severe upper canopy. So the first thing is to try to work out each season, which disease am I controlling, and then which one am I going to time with my fungicides. And the way this works is actually quite simple. So if we're sowing, and I put the word germination in for this year because I know a lot of crops got sown and didn't germinate for a couple of months. So if we're sowing and germinating early in the growing season, our plants are becoming established in this April-May period. And what really happens then is that the spore release in blackleg is really only severe during the winter. So it's the June, July, August period where the blackleg is severe. So if we can get our crops sown in the ground early under warm conditions, they establish early and they basically become established before the disease turns up in the winter. And if they do that, we generally get low crown canker. And this has been, in my opinion, one of the real reasons why we've actually been able to control blackleg quite well in Australia in the last 20 years, because we've moved our sowing window earlier, about 30 days, and that's meant that we establish our crops before the winter and before the blackleg becomes severe. So that we know that after about the third leaf growth stage, any infection that occurs after that won't result in plant death. And after about the fifth to sixth leaf stage, you won't even get any more crown canker after that. So, which is why our seed treatments and our foliar fungicides, we always concentrate on controlling the disease in the seedling stage, and that then results in low disease at the end of the year. On the flip side, with our upper canopy, if we sow and germinate early, it means we flower early as well. And what we saw 20 years ago was our crops were already coming into flower late August, early September, and by that stage, they missed the blackleg infection period to some degree. Whereas now, a lot of our crops are flowering in late July, early August, and they're still in that winter period and getting that infection. So in some ways, we're sort of damned if we do, damned if we don't. If we sow early, we miss the crown canker, but then we're still flowering in that infection window and we get upper canopy. On the flip side, if we sow late Our crops are staying vegetative in that winter period with the blackleg's active. We get lots of crown canker, but then they flower later in the year, end of August, early September, and we get no upper canopy infection. So that's really important to understand. Which disease are we controlling this year? So how do we go about controlling blackleg? As I said, I've been asked to speak a little bit about genetics, and so I've got a couple of slides here just on the absolute basics. But in canola, we have two different types of disease resistance in our cultivars. We have the major gene resistance, which is very, very well understood. And the way this works, it's called a classic gene for gene interaction. So these two photos here I took last week in our disease nursery. So this is canola on canola, different cultivars, different genetics, exactly the same inoculum, exactly the same disease pressure. And you can see here, this cultivar has an effective major gene. So the way this works is a spore lands on the plant. The plant recognizes a particular protein in that spore, turns on its defense mechanism, keeps the disease out, and it's immune. So it's an effective major gene. This plant here still has major genes, but the pathogen has overcome those genes. And the way the pathogen does that is it just mutates slightly so that protein that the plant was recognizing is no longer recognized. And therefore the plant doesn't turn on its mechanism, and the plant then is susceptible to that particular isolate. So all cultivars in Australia have these major genes. Some are effective, the pathogen hasn't overcome them, and some are ineffective where the pathogen has overcome them. And that's pretty much what you need to understand with a major gene. Now, this is some good news too. So in Australia with our GRDC project, we have 32 monitoring sites in the 4 canola producing states, and we sow cultivars which contain those major genes side by side. And each year we assess them to determine which genes are effective and which genes aren't effective in different regions. And this data is now in the blackleg management guide as well. So you can have a look at this table. As I said, it goes for all of Australia, and you will be updated each year. But what you can see here is these are the letters which represent the major genes in our different cultivars. You can see the B's and the C's. These were the major genes which go back to in the 1970s, and A was introduced about 25 years ago. But you can see these have all been completely overcome, and they're no longer effective at all in Australia. So they were effective when they were first introduced, but they're not effective anymore. And this is, I guess, is where it gets more useful, is like group H. It's our latest major gene introduced a few years ago, and you can see in most sites it's completely effective. So any cultivars which contain a H in their letter means that they're immune to disease at that site. You can see here there's one at Hamilton where it's not, and this is because the group H was originally brought into Australia in winter canolas, which we use for grain and graze. So where those cultivars have been grown for a number of years, they have been overcome. But in other regions where the cultivars with the group H have only been released recently in the spring types, they're still totally effective. So we expect over the next couple of years, we'll see a few more red cells starting to pop up. But at this stage, the group H generally is very, very effective. But then you can see some of these others like the group Ds, ineffective at some regions, ineffective at others. And that's really a result of where the cultivars are grown. So if people are growing group D cultivars, they become ineffective. If they're not growing group D cultivars, then they remain effective. And group F is a little bit the same. The interesting one in this table is the group S's. So for those of you old enough to remember, that's the sylvestris resistance, which was completely destroyed 20 years ago and hasn't been really grown since. And now there's a cultivar come out with an S back in it, and we put it back into our monitoring. And you can see, because it hasn't been grown for 20 years, the blackleg populations have actually changed, and that group S is actually looking quite effective again. So this is a great place to start. When you look at your cultivars, you know what cultivar you're going to grow, look at its resistance group, have a look at this chart, and then say, well, is it likely to be effective or not in my region? The other way you can then check to see is it effective or not is by just walking out into your crop. So as I said, this major gene resistance keeps the disease out. So if it's effective, you won't get any leaf lesions. And it's very easy to see, clean cotyledons, clean leaves. And if it looks like that, you've looked at your cultivar, you know it's a resistance group. You've looked at the chart and the management guide, and it says it should be effective, and you've checked in your crop, you can be certain that it's effective and you won't get any benefit from applying a fungicide. If you see the leaf lesions, that means that major gene resistance has been overcome on your farm, and then you need to look at other steps to protect yourself from the disease. Now, the other resistance that we've got our cultivars is the quantitative resistance, which is actually the main, I guess, the backbone of our resistance in Australia. Because I said these major genes get overcome very quickly, and when they do, they're all or nothing. So they're immune one day and totally susceptible the next. So all our cultivars now need to have what we call quantitative resistance in combination with that major gene, so that when the major gene gets overcome, our crops don't just die, they fall back onto this quantitative resistance. Now, the quantitative resistance we call a partial resistance, so it's not an all or nothing, but you'll get various levels of disease. It's poorly understood, this genetics, look, there's a lot of work being done at the moment. It's becoming a real major focus to try and understand this quantitative resistance and better use it much more strategically. But at the end of the day, I guess the way the breeders do this is they sow all the germplasm, canola on canola, and they go through and keep cutting plants that look clean and throwing away plants that look like they've lots of disease. So they're always selecting for this quantitative resistance. We don't understand the genetics, but we do know that it's additive. So if you've got more genes, you're more likely to have, good resistance, and fewer of these quantitative genes, you've got less. The thing with quantitative resistance, if you want to look how effective your quantitative resistance is, you have to wait to the windrowing stage, and then, or straight after windrowing, when it's easy to walk through your crop and cut plants open and have a look to see what the quantitative resistance is doing. It's not related to leaf lesions. So ignore your leaf lesions. Leaf lesions in quantitative resistance is more of an indication of how severe the disease this year. It's not a good indication of how effective your actual resistance is in your cultivar. So the other thing with quantitative resistance is because it's not an all or nothing, it changes with the disease severity. So for instance, I've just got here, we've got 3 different levels of disease severity, with our high, moderate and low. High being lots of rainfall, canola, wet canola rotations. And this is really the environment where we see the big yield responses to fungicides. It's really the environment where we see the resistance being overcome, the pathogens evolving and mutations occurring. In these other regions, we see those processes to some degree, but much, much more reduced. So if you're in a 1-3, 1-4 year rotation in a moderate, growing region, rainfall zone, we don't see the massive blackleg issues that we can in these other regions. That being said, we can still see very high levels of disease in those really, really wet years. But in a normal year, in a normal situation, it's often hard to get yield responses to fungicides in these 2 regions. So for instance, here's a bit of an example. If you've got a moderately resistant cultivar, if you're growing in a really high disease pressure, you can still get reasonable levels of disease and still get a benefit from fungicide. Whereas if you're an MR in a more moderate zone, it's unlikely that you'll get a benefit from a fungicide. But like I said, if it really get a lot of rain, well, then it may actually look more like this, and then before you do. So you've got to play, I guess, the seasons off a little bit, but it's good to have that base ground knowledge to know where am I starting. And if you're in a lower rainfall zone, you can grow a lot of resistance types and really not expect to get much yield loss at all. Now, one of the things with quantitative resistance is it has been in the past believed to be robust. So the quantitative resistance is what it is and doesn't change over time. So the work that GRDC has been funding over the last few years has shown that that's actually not the case, that the quantitative resistance will be eroded over time. And we've seen this really anecdotally over the last few years, where cultivars which have been really high yielding, for instance, we've all grown them, those hybrid group B cultivars, which have had really good resistance, but we've seen over time they get more and more disease, and they respond more and more to fungicides. So in this current project, we're trying to set up a system where we can actually monitor and actually have a look at how much disease is actually occurring and actually get some real objective data on this. So we've got agronomists sending stubble of cultivars for their region, which they think have lost resistance. And then we've screened those stubbles here in Horsham against a whole range of different controls, and then just looked at how they react. So, for instance, this cultivar here, its blackleg rating is an RMR. If it's actually performed better against that stubble that the agronomist has sent in, we give it a green, and if it's performed worse, we can give it an orange. And you can see when you look through these cultivars, you can see that in some regions, the blackleg population isn't very virulent against that particular cultivar, and they actually do better than what their blackleg rating is. And in some regions, they do a lot worse. And this, I guess, is taking, our management to a next level where we hope that with this type of data, we'll actually be able to give you much more regionally specific information about how your quantitative resistance is performing as well. Because obviously, if you're growing this cultivar, there's a very big difference if you grow it in Esperance compared to if you grow it in Cowkey. In Esperance, you wouldn't need to put a fungicide on, but in Cowkey, you definitely would. So having this information is really critical. It also means if you're in Cowkey, you may actually be better to change cultivars. Whereas if you're in the Lower East Peninsula, you say, yeah, I can still manage that. I'm just going to put a fungicide on, etc. Some really interesting things here too. For instance, some of these Group C cultivars, which haven't been grown for a couple of years, they've dropped out of favour. The blackleg population's changed quite dramatically. And you can see here they actually do a lot better than what their blackleg rating is. Whereas some of, like I said, some of the really common Group B hybrids, in some situations performing a lot poorly against those particular populations. So this data here is really good. Really pleased that it looks like we'll be able to actually measure it, that people will be able to send us in stubble, and then we'll be able to tell them whether their stubble has evolved to overcome their quantitative resistance or not. So over the next couple of years, we certainly hope to be able to include this type of information into the blackleg management guide. But at the end of the day, if you're feeling confused at all about genetics, the easiest thing to do is at windrowing growth stage, just go out and check your crops. If your crop is looking like this, you've got lots of disease, you've got yield loss occurring, and you want to change your practices, whether that's changing cultivars or distances to stubble or putting fungicides on. Whereas if your crop's looking like this, obviously continue your current practices, everything's good. So I think a lot of blackleg management is actually just checking, having a look at the end of the year, how did I go, and then knowing whether you want to change your practices or not. So moving forward to where we are right now, August 2024, and it's actually a very difficult situation because we've got crops which are at cotyledon growth stage, and in fact, we've probably got plants still emerging with each rainfall event. We've got crops which are vegetative, we've got crops which are running up to flower, and this is probably around this stage where the majority are, but I'm certainly aware that we've got crops, in full flower as well. So we've got, from a growth stage perspective, we've got the full suite of growth stages. And as I said, a few minutes ago, this really affects what disease you're chasing, because if your crops are looking like this now, you have to protect them because it's still August, there's still disease around, there's vulnerable to crown canker. Whereas if your crops are like this, they're obviously vulnerable to upper canopy, and you need to protect them for the flowers. If your crops are in between, you've probably escaped all disease. It's too late for the crown canker to get them, and they're not mature enough to get the upper canopy. So you're probably pretty safe. The other thing with this year is that there's reports of massive differences in the amount of disease. So in Victoria and South Australia, the crops that I've looked at, they all look like this. They're all perfectly clean. In fact, I'm going to say that 2024 is one of the lowest disease pressure years that I've ever seen. So we haven't had, during the growing season, we haven't had large rainfall events where things have stayed wet, and blackleg being a fungus needs that constant wetness. We haven't had constant leaf wetness, etc. And the amount of disease we're seeing is very, very low. And I don't expect to see much yield loss in Victoria at all. Whereas I know we're hearing reports now from Western Australia after a month of very wet conditions that blackleg has really come in and is really quite severe in some situations. So I guess from a management perspective at 2024, it's very important to work out, am I going to be spraying my my petals and my plants, my flowering plants, and protect them from upper canopy, or am I here and I'm just going to be looking after my seedlings and worrying about crown canker, or am I in between somewhere? And really, actually, there's no point in me spraying at all. And how much disease have I got? Am I in a Victorian situation where there's no disease? Doesn't really matter what growth stage I'm in. There's no disease. And if you look at the weather forecast, there's no rain for the next at least 7 days. So by that stage, we're into mid-August. Things are getting too late for disease. So I wasn't planning to talk about crown canker, but I'm going to give a very quick synopsis on crown canker, given that we do still have crops at cotyledon stage, unfortunately. And in fact, I'm aware that some crops have got cotyledon, same flowering plants in the same crops as well. So really, in summing up for our crown canker spray decision, it's what is my disease pressure? Am I in a canola, wheat canola rotation, high rainfall, or not? That's the first thing I'd look at. And then what's the growth stage of my crop? If I'm past that 6-leaf stage, don't worry about it. It's already too late if the disease is in there, and if the disease is not in there, you won't get any benefit because the plants have grown through that vulnerable stage. So I'm suggesting that most crops are in that situation where they are past that 6-leaf stage, so there's no point in protecting the cotyledons. And then finally, know the blackleg rating, know the resistance group, look at that chart, look at your crops to see if they've got leaf lesions or not, so you can know if they've got the disease. And really, the best advice I can then give you is to use the blackleg CM app. You put all those parameters into the app, and it predicts how much yield loss you're going to get, and then you can look at your fungicide options there, and it'll give you an economic return. So this app's been used for a number of years now. It's been verified over a period of time, and it's giving really good information on that, on the fungicides and economic return. So, but the things you really need to know is what's my disease pressure and what's the growth stage of my crop, and then the app will give you a pretty good indication of whether it's worthwhile or not putting a fungicide on. I'm not sure I put that one up there. So really now moving on to our upper canopy infection sprays. This is really the decision that most people are going to be making now in August. So upper canopy, as I said earlier in the talk, it's really a result of our crops flowering early in the growing season. And what happens is when conditions are conducive for blackleg, we'll get our flowers infected, we'll grow along a peduncle into the branch, and then it causes the necrosis and blockage of the vascular tissue. And this is the critical stage of where the yield loss occurs. So these stages can occur, and you can see them, and they cause no yield loss at all. It's a little bit like the crown canker. It has to get into that vascular tissue and cause the necrosis for the yield loss to occur. So the most critical thing with upper canopy is when the infection occurs and the timing of the year. So when we get rainfall event and crops are flowering, we'll see the same symptoms occurring, regardless of what part of the year it is. However, the disease needs time, as I said, to grow in and cause the damage. So if we see that level of disease in July, the disease will have time before harvest to grow in, cause the necrosis, and cause significant yield loss. And we've measured up to 20 to 30% yield loss quite easily when we're seeing this level of damage. When we're flowering in the August period, we'll still see some levels of yield loss, but vastly reduced. And then when we get to September, you still see the same symptoms, but the disease just doesn't progress. And by the time the disease is going to progress, you've already harvested the crop. So there's really no point in actually spraying and protecting the crop at all. So a couple of years ago, we released the UCI blackleg CM app. Now, this is really a spray decision tool. It's a very simple app at this stage. Over the next 2 years, we really hope to put a lot more information in there as we get more information. So we're pretty confident that it's going to improve considerably, but this certainly still has this place to play. So in this situation with the app, you fill in all the crop parameters, distance to last year's old stubble, your intensity in the region, rainfall, etc. So it puts you into a high, medium, or low risk situation to start with, and then you put in your date of first flower, and then it gives you all your fungicide options. So in this situation here, the output, this is done from Horsham, we're spraying on the 1st of August and we've got disease in the crop, and it's saying if we don't spray, here's our yield, here's our distribution for our yield. If we do spray, you can see here we're getting quite a big increase in our yield target when we put that fungicide on. Conversely, when we leave everything identical, but now we're going to say we're going to flower on the 1st of September, both charts are identical. We're saying there's no yield loss at all. So, and I think this is the most important thing to remember from upper canopy, is that date to first flower is absolutely critical when you're looking at what's the potential yield loss and therefore what's the potential return on a fungicide. And this is why we get so many, I guess, contrasting results from growers and agronomists, is because the window for, I guess, yield loss declines so quickly. So you can be mid-August and get some yield return, and then 2 weeks later, you flower 2 weeks later and you get no yield return at all from the spray, because it really does cut off that quickly during that August period. It goes from beginning of August, high risk, to end of August, almost no risk. So depending on when you commence flowering in there and the season turns out, is how much yield loss you will or won't get. So the really nice thing that we have now is we have an extra column in the management guide which tells you your blackleg rating for the upper canopy infection. So as I said, this has been a joint effort from the GeoDeseed project and from the seed companies, and you can see here where you get an R-rated UCI, they are cultivars which in this situation have that H resistance, which we believe are still immune in Australia to upper canopy. So if you're immune, if you get an R UCI, you're immune and you don't have to worry about upper canopy regardless of when you flower. If you come out as an MR, that means we think you've got solid resistance, but under the right conditions, you will still get yield loss. In fact, we know you'll get yield loss with that type of resistance. So if you flower too early and you're in a high severity disease situation, you'll still get yield loss. And then we've got another group here, which means that the genetic resistance is quite low. And then we've got a fourth group where we're just saying it's really non-existent. So we've only got 4 groups for upper canopy. They're immune, they're good resistance, they're okay resistance, and they're virtually no resistance. So they're the 4 groups you've got to work with. As I said, we've also got these charts here for whether those major genes are effective in your different regions. So they are certainly work also for upper canopy. So the blackleg resistance now works for upper canopy and for crown canker. So you can check them, and you can check for leaf lesions. So if for instance, you're a group D and you're not sure, you can look here and say, yep, group D is effective in my area. You can then go look and say, yep, I have no leaf lesions, and therefore I can be pretty confident I don't need to put a fungicide on for my upper canopy infection. The fact sheet also gives you a bit of a description of what these UCI blackleg ratings actually mean. So again, for instance, if you're an R UCI, it'll tell you yield loss is very unlikely and chance of getting a fungicide response is very unlikely. If you're an MR, yield losses will only occur if disease severity is high and flowering starts early in the growth season. The big question I guess we have at this stage is we don't have a lot of regional data on these things. So if you're an MR, for instance, at Horsham, where I am, it might mean that if you flower in early August, you don't need a fungicide. But if you flower in, say, south, like at Lake Bolac, and you're an MR, you might still need a fungicide. So really, that's what we're working at now. So that's really what this slide is about. With our current project, we're monitoring 150 paddocks over 5 years. We're looking at all the phenology when the crop's flowering. We're looking at their genetics, what their cultivar is, what their blackleg rating is, what their upper canopy blackleg rating is. We're collecting all the weather data. And then we're spending a lot of time looking at the upper canopy infection symptoms, and we're getting growers to leave unsprayed strips, so we look at the yield responses as well. So over the next 5 years into that UCI app, we're really going to start building in all this regional data so that you'll be able to go, okay, I'm in Horsham, I'm in Esperance, I'm in Dubbo, or wherever you happen to be, and it will then put you into a risk category for upper canopy straight away. And that's really the missing part, I guess, of the app at the moment. And then you'll be able to say, well, if I'm an MR, in Wagga compared to an MR in Horsham or Birchip, what does that actually mean for that resistance, and what is the probability of getting a yield loss and therefore justifying a fungicide? So that's all coming over time. but we are making quite good progress on that. So I guess in summing up, do I right now put a 30% bloom spray on? The first thing is, am I having an early bloom or not? So is my crop still vegetative on the 7th of August and we're not going to see any rain for the next 7 days? Well, then the chance of getting upper canopy yield loss is very low. Late bloom, don't apply a fungicide. That's really easy. If it is early bloom, and for most people I'm saying is it commences flowering in July up till the middle of August, well then fair enough. You can decide whether to put a fungicide on. No leaf lesions. My major gene is effective. I've looked at the charts. Again, I don't need to apply a fungicide. In this situation, okay, I'm early bloom. I've got leaf lesions. I don't have major gene resistance. Then as the agronomist, you or the grower, you need to make that decision on the economics of, how much return am I going to get? If I've got high yield potential, obviously you've got a much better chance of getting an economic return from your fungicide. And then you can fill in the parameters in the app. What is my disease pressure? How far am I away from last year's canola stubble, etc.? What is the blackleg rating of that cultivar? If it's MR compared to an MRMS, for instance, and then make the decision to put that fungicide on. The one thing I've got here, which again is not in the app yet, which we hope to build into the app, is what happens in the spring, which obviously you don't know right now, but it can have a big impact. So we now know that the same levels of disease can cause very different levels of yield loss depending on what happens in the spring. So if we have a spring with a cool, moist finish, plants can tolerate a reasonable amount of disease and not get a lot of yield loss. But the same amount of disease under a hot, dry finish can cause a lot more yield loss. And it's pretty obvious when you think about it, because the disease is blocking the vascular tissue. So if the plant's moisture stressed and half of its vascular tissue is blocked, obviously that has a much bigger impact on the plant, and the plant tends to drop and shut down a lot quicker, and the yield loss can be a lot more pronounced. So but there's more work to be done in that area, and over time we will hopefully build that into the app. But in some situations, it really doesn't actually help you a lot, because you've got to make the decision to put your foliar fungicide on at 30% bloom, and you don't know what the spring will do at that stage. But it's something which I guess you've got to consider as a worst case scenario, and that could certainly be on the cards this year. Looking at the amount of rainfall we're unfortunately getting at the moment. As with crown canker, I really advise people to get out and have a look, especially when people say they do or don't get yield responses. We've now, upper canopy symptoms are very, very variable, but there's two symptoms which reliably result in yield loss. The first is what I just call or term darkened branches. So you can see here, I've got two branches, one at windrowing stage, so this is a sort of 50% seed colour change in your pods. The branches have gone very dark compared to the ones which have stayed green. And this is a classic that the vascular tissue has been damaged, and that branch on that plant is shut down prematurely, and this is where we see all the yield loss. So once you get your eye and you start looking through crops, you can see these symptoms and pick them out very, very quickly. And then you can get your secateurs and cut them and confirm that the pith inside is dark. And that's a really good indication that upper canopy has caused yield loss on that crop. Obviously, there's nothing you can do for the current crop, but if once you start getting your eye in, you will actually it'll give you a really good indication of whether you should have sprayed fungicide or delayed sowing or whatever to delay the flowering period. So it'll give you a good indication whether on your farm whether upper canopy is an issue or not for future years of management. And the other one is to start splitting plant. A pair of secateurs, you can split them or whatever and look for that blackened pith. That blackened pith, again, is a really good indicator of yield loss, because, like I said, a lot of infection for upper canopy may be totally superficial, occurs too late and doesn't cause any yield loss at all because it doesn't get in and do the damage to vascular tissue, and that's really the key to the yield loss. Just finishing up now, there's a couple of other things. Obviously, the fungicides we put on at 30% bloom sprays are very broad spectrum, so they control other diseases as well. So we've noticed that the 30% bloom spray can have a really profound effect on powdery mildew as well. We don't have good data on yield losses from powdery mildew, but I know people who are windrowing and harvesting really don't like it. So I take it as just a bonus that when you're putting a 30% bloom spray on, you'll also get a lot less powdery mildew. And the other one, which obviously everyone is interested in, is sclerotinia. Again, it's very fortunate that the timing for upper canopy, blackleg and sclerotinia are identical, that 20-30% bloom stage. And the reason for that is because it's about as late as you can go and still get good penetration into the canopy. And with sclerotinia, the main thing to remember is it's the main branch or the main raceme which causes yield loss. You can get a lot of sclerotinia on the branches, but because they're only individual branches, the yield loss in many cases isn't that great. But as soon as you start getting that main stem infected, that's where you get the yield loss. So that's why we push this spraying out to pretty much as late as we go and still get good penetration to protect that stem. Thing with sclerotinia is that it's a very, very difficult disease to predict if it's going to occur or not. It's like I've said here, all the stars must align. It's like there's 5 different factors which must all come together, and when they do, sclerotinia is very devastating. But if only if those 4 factors come together, it doesn't turn up at all. So there's very minor differences, and we have some work with GADC at the moment. We're monitoring crops in southwest Victoria, and we can see individual crops which have had, in 2022, up to 45% of plants with sclerotinia, and 2 kilometres down the road where they only had 5%. So we don't know yet why that difference occurs, but we are certainly aware that it does. And it's not just an issue for canola, it's an issue for all crops, and it's an issue for all countries as well. Sclerotinia is incredibly difficult to predict. So in saying that, we do have Sclerotinia CM app. I keep pushing these apps, but I do think they're very good. In this app here, it's again a spray decision tool. So really the way we've dealt with this one, and this was developed by New South Wales DPI and DPIRD, is that you, as the grower agronomist, you have to actually determine what is the threat of this paddock. So you know the paddock history, you know whether that paddock and that crop gets sclerotinia or not in the past, and then you fill that in. And once you've determined that, then it'll predict your yield loss, and then it gives you all your parameters. So it tells you what was the, ask you what the rainfall events were for the previous 3 weeks, and what's the forecast for the next 2-3 weeks, what's the growth stage right now. And then at that point, it is a really good app at predicting what the yield loss will be and what the fungicide returns will be. But the key is that you actually have to put in whether the crop is at risk or not. And most people will know that. They will know whether they get sclerotinia most years or not, or they will know if they're in a region which never gets sclerotinia. The tricky part of this is where you're in that intermediate area where sometimes it turns up, or it turns up 1 year in 5. And you're never quite sure whether it's going to turn up or not, and that's hard. And like I said, we are doing some work to try and get better data on that, and that's the holy grail, is to produce models which actually then predict how much disease you're going to get. But at this stage, we're really not at that point, unfortunately. Now, just finishing up here, I'm just going to talk quickly about fungicide resistance. So in blackleg, as I said, it does overcome our cultivar resistance. We've known that forever. We're now aware that it also overcomes our fungicides. And we have a screen we've been doing for a number of years now. So this is in conjunction with Dr. Angela Vanderwell and her student Alec McCullum at the University of Melbourne, where we take canola stubbles from all over Australia. We suspend them above canola plants, which have been treated with different fungicides, and we let the spores shower down and then look at the effect. And you can see here, just in these photos, here's our untreated, where all the cotyledons are affected and destroyed. Here's where we put our SDHI fungicide on, and we don't see any disease at all compared to our DMIs, which we know have got some resistance to them. So the good news in this is that in all the screening that we've done, we haven't found any evidence that the SDHIs have been overcome. So we are very confident that they're completely effective across the entirety of the country. Whereas with our DMIs, we see a lot of populations which can do this level of damage. They certainly are not, they're still getting some protection in these screens compared to the untreated, but we can pick up a fair bit of damage from them. The interesting thing is we don't yet have any evidence that there's actually field failure for the DMIs. So these screens we're doing are very, very hot, very, very sensitive. So we know there's isolates out there which can do that damage, but we're not seeing evidence for field failure, which is interesting. But the really nice thing with this is, from this data, with the chemical industry, we've put together the crop life recommendations, and basically the way these work is that you go through, so at the seed, when you're planting, whether you're putting a seed treatment on or in furrow, it gives you the options. Have you put on a group 3? Have you put on a group 7? Have you put on none? And then again at the seeding stage, it asks you what you put on, and then it gives you what options that you can then put on for the upper canopy spray at the 20-30%. And really, the take-home message with fungicide resistance is, if you don't need to put a fungicide on, don't. That's the best way to preserve, the efficacy of that fungicide. But if you do put it on, try and rotate the resistance groups, and that's what this chart will, help you with here. For instance, if you put a group 3 on, it's telling you, you can put a group 7 on, and then what options you've got at that later growth stage as well. So obviously, if you put none on, you get more options down here. If you put more up here, you get less options, etc. But you can just work through that chart to look at what's the best practice at this stage. The other nice thing with fungicide resistance and blackleg is that our colleagues at Melbourne Uni have now mapped the mutation in the fungus, which gives them their ability to attack the DMIs. And because they've done that, we can now track it. So they can look at the frequency of isolates which can attack the DMIs at any particular growth stage. And then we can test all these recommendations and actually get real hard data. So instead of just coming up with a bit of a model, we think how it might work, we can actually start testing all these situations and actually get real hard data behind it now. So we've got that technology. So I think over the next few years that these crop life models for how to manage our fungicides will improve dramatically as well. And I guess just in final summing up with the things with the fungicide resistance strategy is that the DMIs, we believe, react a little bit like the minor genes. So you see this erosion of efficacy over time. You don't see it all or nothing, but the evidence to date is that in other crops and other diseases, the SDHIs, they're all or nothing. So when they get overcome, the SDHI will become completely ineffective. So when we do see it overcome, it's likely that in the field, it will be overcome as well. Whereas with the DMIs, even though we're seeing it overcome in our populations, we're still seeing it being quite effective in the field. So I guess that's the danger, and that's what we've got to be really aware of. And I guess that's the risk, that we could lose those SDHIs. But fortunately, at this point in time, all our data shows that they are completely effective and work very, very well. So look, that's all I'm going to talk about today. Very happy to take questions. From an information perspective, get that fact sheet. That fact sheet is becoming more useful all the time. It's got all the blackleg cultivars in there. It's got their blackleg rating. It's got their resistance group. It's now got the charts for the different regions, say which resistance groups are effective in which different regions. And it now also has the upper canopy blackleg ratings in it as well. So it's a great resource, and it's updated every 6 months with all the new cultivars. And when you get to making those decisions in real time in your paddock, I think using these apps is a really good place to go. And I talk to a lot of agronomists who don't potentially use it for every single paddock they go to, but they will just jump on it to make sure they are making the right decisions and not doing anything silly. But if you use them well, they've been verified a lot, and they do predict yield loss very, very well, assuming you put the right information into it. And look, I guess in summary, in most situations, in a lot of regions in Australia, we don't need to use fungicides because the yield severity isn't there. But where you do identify that yield loss is going to occur, the fungicides are actually fantastic, and give excellent results. So I'll leave it there. Good luck with your decision making this year. I know it's a tough year given how widespread the crops are in their development and how much disease they've got. I'm happy to take any questions.
Thanks, Steve. We do have a couple of questions just to wrap up the session. One of them is relating to fungicide longevity, I believe. So the question is, would SDHI fungicide seed treatments have broken down if dry sown and sat for about 6 or so weeks prior to emergence?
That's a really good question, and that's I've been asked that a number of times this year. And to be honest, I don't have any data on that. However, I think everything we know tells us that they should still be effective. That if there hasn't been enough moisture to germinate the seed, there probably hasn't been enough moisture to move the fungicide away from the seed in the soil either. So I'm pretty confident that fungicide is still effective to that seedling. Yes.
Great. Another question that has come through is, with delayed flowering, how late into the season can or will blackleg cause upper canopy infection?
Yeah, like I said, that will vary a little bit on different growing regions. But look, everything we know is so for instance, in Victoria right now, I'm sitting in Horsham, we're in early August, we've got crops, none of the crops around here, or very few of the crops are flowering yet, and we've got no rainfall forecast for at least the next 7 days. That puts us through nearly into mid-August, and I'm going to say, no, we don't need to put a fungicide on. Whereas if you were flowering right now, if your crop's already is commencing flowering now, and you've got leaf lesions on that crop, well then that's where I'd be going through the process and looking at the blackleg rating, etc., and determining to put a fungicide on. So certainly once you start flowering past mid-August, the disease risk reduces dramatically, and by the time you get to September, it's almost non-existent. So we're in that real transition phase now. But also remember, you need to have rainfall events during that time to actually, get the infection. So if you're not seeing any rain on the horizon at the moment, and you're not sure, I would be probably holding off putting a fungicide on until you do get those infection events.
And Steve, I'm going to finish just with 2 recap questions. The first one is, you mentioned fungicide decisions. So should we still be using our group 3, so our DMI fungicides?
Yeah, look, at this stage, until we start seeing field failure, I'm totally happy to keep using the DMIs. And like I said, I think it's the critical point at the moment is do I use a fungicide or not? And then secondly, I want to be able to rotate my fungicides. And therefore, if you're going to rotate fungicides, the DMIs are a critical tool to be able to do that. And while we don't have field failure, I think that's a really good way to go, because it really then does prolong the life of our SDHIs, which, as we all know, are the backbone of our, our blackleg management at the moment. So yeah, I'm totally happy to keep using those DMIs.
And Steve, just remind us once more, where do we get the new blackleg ratings for upper canopy? Where do we get those from?
Yes, if you just Google Blackleg Management Guide 2024, it'll come up straight away. It's on the Geodesy website.
Fantastic. Thank you, Steve, for this incredibly informative webinar. I'm sure it's provided all of our viewers with the confidence going forward in this season. I would also like to close by saying a huge thank you to Bridget and David at Ag Communicators for organising this AFREN webinar. As we bring the session to a close, I just want to ask everyone to not exit just yet. A quick survey will appear on your screen, and we would greatly appreciate your feedback on this session. So Steve, thank you once again. And from all of us at AFREN, we want to thank all our viewers for joining us. Take care, and we hope to see you again at a future AFREN event.